Okay. Yeah, okay, so welcome. My name is Sczyrba. I will talk a little bit about the theory about genome assembly, because the next step we are going to do is assembling the genome out of a reads. Right? So far you were working with the raw reads or the trimmed reads and so on and looking at the reads. And now we are actually going to assemble the genome out of it. And just to give you an idea of what's going on in the next steps and what to expect from such an assembly, I have a couple of slides summarizing the general problem of genome assembly, basically. So when you look in the past, there were basically different strategies how to assemble the genomes. And the first strategy was actually an ordered shotgun assembly, right? And this is what you all know from the first human genome. There was this huge human genome project where, in an international kind of project, a huge physical map was built. And then using the physical map, you were basically sequencing the clones, right? Clone by clone. And this is called an ordered shotgun assembly. And then at some point, well, basically there was a company at that time, Sczyrba, that decided to go a different way and to do a whole genome shotgun assembly. And this is actually what we do nowadays, whole genome shotgun. And the nice thing about the whole genome shotgun is that a large portion of the sequence is obtained rather quickly. So you just sequence it, you assemble it, and most of the sequence you actually get assembled. The only problems are the repeats with this kind of approach, and you will see this later. And then, of course, there was next generation sequencing, and there were all these short reads. And that's and for the short read assembly, people had to invent basically a completely different assembly strategy. So here's the classical way. Again, this is what has been done in the human genome project. You start to do genomic DNA, you build back libraries, you organize the libraries in a map so that the map actually spans the whole genome. And then you take each back and then you do shotgun sequencing of each back. You end up with an assembly of the back, and then based on the map you can reconstruct the whole genome, right? And as I said, it's a huge effort to actually get the map constructed before you can actually sequence, okay? But what you end up with is this here. And that's also, and this is for the whole genome shotgun approach. You just leave out this middle part here, right? You just go for the DNA and do shotgun of the DNA, just fragment the DNA and sequence all the little fragments and then try to reconstruct it using a computer, basically. Okay? And the way it works, just to get some terminology here, so you start with the reads down here, right? From the reads you assemble what's called a contiguous sequence. That's basically a consensus of all the reads you sequence. And then you can do a step which is called scaffolding using different approaches. You can either use read pairs, mate pairs, by doing paired end sequencing. You have an idea of how the contigs are actually ordered, right? And then, of course, you can try to close the gaps if you like, but you don't have to. What you get is what's called a scaffold out of it. Right. Okay. And then and one thing I want to talk about is this statistics here, right? Because the question is always like, how much do I have to sequence, right? If you think about Illumina sequence, question is, well, how many reads do I need to close the genome, right? And that was a question very early on, way before the human genome project. And that's a paper from 1988 from Lander and Waterman. And they came up with statistics to actually get an idea of how much sequence you need to close such a genomes, right? And it's called the Lander-Waterman statistics, basically. So what is the statistics about? Well, as you all know, probably there's a relationship between the reads and the number of contigs, right? If you only have one read, you have one contig. And if a read is long enough, you have the complete genome, right? If you have very short reads, you need a lot of reads, a lot of coverage to actually reconstruct a genome. And for this whole statistics, the idea is that the reads don't have any errors, right? So it's a very simple statistics. No errors in the reads. And these are all the numbers you need. You need the number of reads you sequenced, the read length. So it depends on the read length, obviously, on the genome size, right? And then you can calculate like all the nucleotides you have sequenced, which is just the number of reads times the length of the reads. Or you can calculate the coverage, what's called the coverage, which is the total number of amino acids you sequence divided by the length of a genome, right? And I did some calculation here based on the Human Genome Project. So if you say, okay, I need 5x coverage of a human genome, 5x coverage means 5 times 3 gigabases, divided by 500. I put 500 here because this is a Sanger, let's say this is a Sanger read at that time. So I need 30 million reads to be sequenced on a Sanger machine, right? If you do this on the Sanger technology, Sanger machine can produce about 3,000 reads a day. So if you do this on a single machine, it takes you about 80 years to just sequence it. And of course, all of you know that 30 million reads nowadays is not a problem at all. And even 500 bases, you almost reach 500 bases with Illumina technology, right? But at that time, it was pretty big effort to do so. So the statistics is now about like how much sequence do I need? Is 5x coverage enough to close the whole genome? Okay. And if we just assume that all fragments are randomly distributed, like the sequencing fragments, randomly distributed across the genome, right? You have a uniform probability in a given interval, and then you can do some calculations. And basically, what you have is a binomial distribution, and if you have a binomial distribution, you can approximate this by this Poisson distribution. Right? I will not go into all the details here, just to give you an idea. So this is what we need. This is a Poisson distribution, and these are the numbers I just showed you earlier. So you have A, which is the coverage, and N is the total number of bases you sequenced, L is the length of the read, and G is the genome size. So it depends on these parameters, basically. The length of the sequence, the read, basically, the length of the genome, and so on, and the coverage. Okay? And now this formula on the left is the probability that K reads start at position X. So suppose you have your genome and you want to know how many reads start at a specific position, right? And you could do the very simple math and just calculate what the probability is. If we want to close the genome, then the K should always be at least 1, right? So at each position we should have at least 1 read starting. Okay, so if we want to know how many gaps we have, right? We just set the K to 0, and then this is a number of gaps we have. Like how many positions are actually not sequenced, right? And you see it depends on the coverage A. Okay. The total size of these regions is just this number divided by the genome length, right? Because this is a probability that a certain position is not covered. And then if you want to get the size, then just multiply this by the genome length. And you can do the same for the number of gaps. So what do you think for the human genome? Like how much coverage do you need? If you go, let me look at the next slide. Let's say it's a Sanger sequencing, 500 bases. How much coverage do you need to close the genome? Any idea?
8 to 12.
8 to 12x. Anybody? 50. Okay. So let's see. 8 or 50. So that's the table here. So this is the coverage, right? So let's say 8. This is the number of reads you sequence. This is 48 million. No, 48 million. this is the number of nucleotides you get, and then this is the number of gaps, basically, right? Because this is the number of contigs, which equals basically the number of gaps, right? So for coverage of 8, you still have 16,000 gaps, right? And your mean contig length would be 186 kilobases. Okay? So 20 to, now what do you say, 50? It's not on my slide, sorry, but you see if you go up to 18, something around 18, you actually get the whole genome, in theory, right? And of course, it's not a single chromosome, so, but anyway, it's just a statistics. Okay, so for Sanger sequencing, 18, something like 18x should be fine if you don't have any errors in the reads, right? Of course, if you have errors, you need higher coverage to correct the errors somehow. Okay, so now let's go down to shorter reads, 100 base pair read length. But much higher sequencing coverage, because now we have this is a bacterial genome, small example, right? So the genome is only 3 megabases. So if you want to close a bacterial genome with Illumina sequencing, you need something like 13x coverage, in theory. Okay, and for like whatever, Oxford Nanopore or PacBio sequencing, the sequence length would be 5 kb on average, right? And now you need something like, well, still, well, how do we go? Now here, something between 8 and 9x coverage. Okay, so that's just to give you an idea of how much sequence depth you actually need to close it. But then, of course, there's not only the sequencing depth, there are the errors you have to correct and so on, and there are other problems like the repeats. So the way you can go in assembly are basically these 3 types of paradigms for assembly. You can do greedy assembly. Greedy assembly would just take a read, map the next read to the end, just see if another read maps to the end, extend the contig, map the next read to the end, and so on, and you can extend it. And it would just take the next one that matches. This is why it's greedy, basically, right? And you don't use any other information. And you already guessed that this doesn't work for whenever you run into a repeat, it doesn't work anymore, right? Because you cannot extend it, or you extend it in the wrong way, basically. Okay, so then the next thing would be, and this is the classical way to do the assembly, is overlap layout consensus. Right? I will show you in a minute how this works. Basically, you identify all pairs of reads, organize it in a big graph, and then using the graph, you reconstruct the genome. Overlap layout consensus doesn't really work for short reads because you have too many reads. And this is why people came up with the De Bruijn graph data structure, which uses k-mers. You heard about k-mers before. It's a different kind of way how to find overlaps, basically. And this can handle like very large data sets and very short reads, right? Okay. So let's go like a quick run about overlap layout consensus. So for the overlap layout consensus, first phase is the overlap. So you have to find all the overlap between all sequence pairs, basically, right? This is n over 2, the number of overlaps, n over 2, which is basically n square, right? If you have 1,000,000 reads, you have to do 1,000,000 times 1,000,000 calculations because you have to compare each read against each other read. This is a lot of computation, so I did a small calculation again. I just checked the human genome paper from 2000, I think, 2001. This is a Sczyrba assembly. And they generated 27 million Sanger reads. And if you do this for 27 million reads, you end up with 10 to the 12 overlaps you have to calculate. If each alignment takes you a millisecond, you end up with 27,000 hours. And if you do the math, then you end up, I think this is something like 3 years on a single CPU, right? Which means you have to parallelize the whole process because you cannot wait 3 years to do one assembly and then you end up, and then you probably want to rerun the assembly. So this definitely has to be parallelized somehow. Okay. And this layout phase is very memory consuming because you have to keep all the layouts somehow in memory. You have to build a graph in memory, right? So what you're looking for are these types of overlays or overlaps basically of the reads, right? You can have overlaps like this. You can have overlaps which are reverse complemented and so on. And each of these overlaps have to be held in memory. And that's a huge graph. And if you have 10 to the 12 overlaps in theory, you would need 1 terabyte of RAM to just construct a graph for the overlap phase, and then using the graph, build the layout, basically. So that was, at that time, another problem. Nowadays, it's not a problem, and not, well, so much anymore, because a terabyte of RAM is, let's say, is easy to get. In 2000, that was almost impossible, because there were no machines that could even address more than 32-bit, right? So there were no CPUs coming out, and so they could actually use more than 2 gigabytes of RAM, right? At that time, 2 gigabytes was the maximum you could use the RAM. Okay, so you see that there are computational challenges to just do this kind of assembly. And how does the layout graph look like? So this is an example where you have different reads, right? And there are overlaps between the reads. So this means it's a reverse complement kind of overlap, and the overlap length is 320 bases of these 2 reads. Here the overlap is only 40 bases and so on. And using all these overlaps, you can build this kind of graph, right? So if you look at this example, F5 and F1, that's this one here. So these 2 reads overlap with minus 50 because here F1, because you have a reverse, right? One of the reads is reversed. This is represented by a negative number here. So you have an overlap of 50 bases between these 2 reads, overlap of 40 between these 2, an overlap of 320 and so on. And you can represent all the overlap you see in the top in this graph. And now the computational challenge is to actually find a path through this graph, because a path, or to find a path that visits each of these edges exactly once, right? You want to use each read once, basically. And if you do this is in computer science, it's called the Hamiltonian path problem, and it's an NP-complete problem. And I don't know if there are computer scientists in the room, but NP-complete is a very hard problem, right? It's very hard. You basically cannot compute it, right? You have to use some heuristics to actually solve the problem, because it would take forever to compute, right? Okay, but anyway, so using this graph and finding a path would give you a layout like this, right? This is the red path, basically. So some of these overlaps are not used in the layout here, and then you end up in this layout. And then using the layout, you can build some consensus sequence, and you end up with a genome sequence at the end. Okay? Okay, and as I mentioned before, of course, there are problems, and the main problems in the whole process are the repeats. Right? Because if you have a genome that looks like this, you have this red part here is a repeat, which is exactly the same in these 2 positions. And when you build the overlap graph, it basically looks like this, right? You have an overlap between A and X, and you have an overlap between X and C, and an overlap between B and X, right? And now you don't really know what the correct order of the different pieces are, because if you only have the graph, you see there are loops in the graph and so on, and sometimes you don't even know the direction of this piece. It could be a repeat, very simple repeat, where you can even reverse this, right? So it's not easy to actually come up with a correct sequence. So that's one thing. And the other thing is, well, the read length is also important for the problem, right? So this is showing an overlap graph for E. coli, basically. Each of these dots are the contigs or reads, whatever. And whenever there's an overlap of at least, these are reads of length 50. So each of these dots are reads of length 50 bases. And whenever there's an overlap between two reads, you have these errors or edges in between. Right? And the graph looks like this if the read length would be 50. So you see many kind of repeats which give you a graph which you cannot resolve, basically. Right? If you increase the read length to 1,000, then it looks much nicer. Right? It basically means there are only, I don't know, let's say 10 repeats of length 1,000 or longer in the E. coli genome. Right? And then you end up with a graph like this. But still you cannot build the final sequence because you don't know which path is the correct one, right? Because you could go from here to here, this way, but you could also go this way or this way. So you have different paths through the graph, and you build different genomes out of it. And only one of it is correct, right? If the read length would be longer than the longest repeat, so in this case, at least 7,000 bases in E. coli, then you end up with only one contig. So the longer reads, the easier the whole problem, let's say. Okay? And that's, of course, why we are looking for long read technologies, because if we get reads that are that long, then the assembly problem is much easier. And how can you use this? Well, when you have a long read, that could be a long read here, this one, right? Well, let me this is a repeat in the middle. This is a repeat, right? And you don't really know if the correct sequence would be ARB or would be ARD, right? Or CRB or CRD and so on. And only additional information would help to resolve this. So if you don't have long reads, but at least if you have these long fragments and you sequence only the ends of the fragments, you would get these mate pairs here. And the mate pair would tell you, okay, A goes together with B, right? Because this read maps to A, this read maps to B. You know the approximate distance between the reads, and you know that this must be the correct assembly. When you look at nanopore reads, well, you hopefully get reads which are longer than the repeat, right? And then you would have a read that goes from C, spans the whole repeat, and continues in D, and then, of course, you know what the right structure is of your genome. Okay? Okay, and then, of course, there are millions of assemblers out there, right? So there's a cellular assembler, which is the assembler that was used to assemble the human genome. There are other assemblers that use a different approach. So this is an overlap layout consensus assembler. These are actually all overlay layout consensus assemblers. Then there are also some short read assemblers and so on. Actually, I think these are all built for long reads, right? And you see there are lots of assemblers. And then, of course, the question is, and these are actually these are de Bruijn graph assemblers built for smaller reads. And then, of course, the question is, which one is the best assembler, right? Which one is the one to use? And what people do is they develop a new assembler, and then they benchmark the assembler against reference genomes. And this is actually something we are sort of doing today also, just to look at these results. Because what you can do is you can take two assemblers, basically. You assemble your reads. And if you have a reference available, you can just map the assembly back to the reference and evaluate your assembly, and then you know which one is a better one. And we will see in a tool to do this later, and you get an idea how this works. So for the long read method, there are basically different ideas how to use the long reads. You've seen already that the long reads are pretty error prone, right? And sometimes it's not really clear if these, for instance, these two reads really overlap, because there are too many errors in the ends of the reads, right? So you have to allow some errors, and you do a realignment. You cannot just look for exact matches, right? Because there are too many errors. But if you allow too many errors, you don't really know if these really belong together or if it's just by chance because you allow too much errors or too many errors. So one way would be to take the long reads and add another data set. So you can add an Illumina data set. And then you map the Illumina reads to the long reads, right, to correct the errors in the long reads, and then basically build the assembly out of a long read. That's one way to do it. Right? And at the beginning, people did it this way. They took the long reads, mapped the Illumina reads, corrected the reads, built the assembly. A different approach would be to start with the Illumina reads. Right? You do an assembly of the Illumina reads. You end up with this huge graph again. Right? And then from the graph, you take the contigs, at least the contigs which are unique. So you extend the contigs up to the point where you have to decide if you go in one direction or the other, which would be a repeat. Right? So you generate the contigs. These are the contigs down here. No, these are the contigs here, 1, 2 contigs. And then you use the long reads to do the scaffolding of the contigs. Right? And then you end up with a consensus sequence, basically, again. Okay? This is done based on the contig level. So you build the graph, you take the Illumina reads, you build the graph, you generate the contig sequences, and then you map the long reads to the contig sequences. A different way would be to take Illumina data, build the graph, and then map the long reads to the graph, actually. Right? And then using this information, build the contigs from the graph. These are just different approaches how to use these kinds of data. And these were the approaches, well, this paper is from 2015. So this is what people did at the beginning when these long reads became available. The assembler we are going to use is Kanu. This is a long read assembler. Kanu is actually built from the Sczyrba assembler. Right? There's this one here, right? The Sczyrba assembler. So it's an overlap layout consensus assembler built for the Human Genome Project. They took the core of this assembler and then basically rebuild it to assemble long reads. Okay? And there are 3 different steps, and you will see these 3 different steps later when you do the assembly. So the first step is to correct the reads. And correcting the reads is actually based on like aligning the reads to each other, not using any Illumina data, not using any extra data. This is just Nanopore data. Right? So what you do is, you have to find overlaps, but finding the overlaps is much faster now because there are different, let's say, different algorithms compared to the Human Genome Project. You do this on the k-mers, basically, hash tables and so on, which are very fast kind of comparison tools, let's say. So you find the overlaps, and then you try to overlap the sequences, and when you overlap many sequences, you can correct errors already, right? And then you look at these overlaps, and you can trim the reads. So you could trim down reads which don't have any overlap to anything else, right? Or you can split reads based on all of these overlaps, just to get higher quality reads. And once you have these higher quality reads, you actually assemble it, right? And the whole assembly, again, is done by the overlap layout consensus paradigm. Okay. Okay, I think I can skip this because we're not doing any well, maybe not. Okay. Well, now let me show you this also. So here's the difference between an overlap layout consensus graph, or the overlap graph, and the De Broglie graph. Right? This is used for short read Illumina assembly. So if you think of like Illumina reads here, right? And this is a very small example where the k-mer length is 3 bases in this case. You can build a graph out of this where you say, okay, I'm looking for overlaps of length, or I'm splitting my reads into these k-mers of length 3, right? So you start with a third read, GAC, and then the second k-mer would be ACC, right? And GAC and ACC overlap with AC, k minus 1 basically, right? And then you take the next k-mer, CCT, CTA, TAC, and so on, right? And you see that for the different reads, the k-mers are actually, some of the k-mers are actually the same, and they end up in the same nodes. So like this CTA from this read basically ends up in the same node as this one here, from a second read. So you have a representation of the genome up to a point where you have a repeat, right? Because here it splits. So you have different reads that don't, that only overlap at the beginning, let's say, but are different at the end, right? So so when you look at the sequence, you have to read it like GAC and then always the last character, GAC, so which is somewhere here, ACAAAGT, right? So up to here, all the sequences are the same, like all of these is the same. And here they actually split in two different paths. So here these continue with TAG and these with CCG, right? And this is represented in this fork in the graph. Okay? And so up here, this is an overlap graph. Overlap means here at least five bases. And when you look at overlaps, then you see that R1 and R2 are actually overlapping with 5 bases, R1 and R3 also, and so on. And then the graph looks like this. Okay, when you have these graphs, you have to correct the errors, right? And the errors are, well, basically 3 types of errors. You have these tips, which are caused by errors in the sequence, and then you get a fork. Bubbles, where you have also different k-mers, right? A bubble would be a sequencing error in one of the reads. So you can think of like this base here being wrong, right? And then you have a path with many reads. This is why this is a thick arrow. Many reads go this way, and only 1 or 2, or at least definitely less reads go this way. And you know, okay, this must be actually the correct one. And the other one is probably just a sequencing error. And down here, this is actually the repeat problem again. And then if you assemble this using a k-mer, and this is basically showing the same as before with the other graph. This is using a short read assembler now, k-mer length of 51. This is not E. coli, but Salmonella genome. The whole graph would look like this, right? So there are many, many repeated 51 mers in the genome. Okay, and as we said, okay, you can increase the k-mer size, 61, and it looks much nicer now, right? You can even increase it more, 71, and now you start, well, maybe it looks like a genome now. And you can even go larger, 81. But if you go too large, then there's no overlap anymore, right? And then everything falls apart. And then the question is, of course, well, which one is the right k-mer size? Okay? And it's difficult actually to figure out the right k-mer size. You don't really know from just looking at the reads. You don't really know the right k-mer size. And what these assemblers do is they use many different k-mers, right? And this is shown here. This is one example. Is IDBA assembler. What you do is you build the graph using a pretty small k-mer, let's say 21. From the graph, you build contigs. Right? And then you take the contigs and you do the whole process again, but you use a longer k-mer size, 31 now, right? And build a graph again. But add to this graph the contigs you assembled in the first run, right? You add it to the graph as if you would have longer reads. And you do this with a k-mer size of 31, 41, 51, 61, 71. And then at the end, you end up with a very nice assembly. And this is usually what the different k-mer-based assemblers do, where they iterate through these different k-mers.
I don't really understand why it all falls apart when we have large k-mer sizes. I don't get
this one here?
Yeah. Yeah. When we have 91, why we don't have any overlaps?
Well, because you have errors in the reads. If you wouldn't have any errors in the reads, then everything would be fine. Right? But because of the errors in the read, so you can think of like having one error in 100 bases or so, then of course everything falls apart, right? Because you don't find any overlaps anymore. Because these k-mers are always perfect matches, right? They don't allow any errors. Yeah. Okay. Okay, yeah, that's it. That's the basic idea of the different assemblers. As I said, we are going to use the CANU assembler, which is an overlap layout consensus. assembler. But there are actually assemblers coming out that use these Kämer and de Bruijn graph paradigms for nanopore reads also. That fly is such an assembler. So when you start reading papers about how to assemble nanopore reads, then there's definitely Canu and there's also fly. So at least if you believe the benchmarks in the papers, then the fly is also a very nice assembler. Okay, so what we are going to do now is we take our